We're now going to move on to two talks by funders, hopefully be of interest to people. So the first one is from Dylan Bonfils and also Claire Connellan, and they're from ISIDORE, Europe. I'm going to tell you about their funding models.
Hello, everybody. Thank you for having us here today. I hope you enjoyed the presentation and the barbecue last night. And we are going to try to make this quick so we can go to the next presentation and the coffee break that everybody's waiting for. So some of you might remember the last year we were there in Cambridge to present to you the ERINHA project back when we were starting to launch new calls for proposal. So this year, after a year and a half of implementation, we are going back to present you some of the results, preliminary results, and the way the ERINHA project works and how we can improve pandemic response and preparedness in Europe. So first, let's have a look at ERINHA. What is ERINHA indeed? Some of you already know ERINHA is a non-profit association with 10 members based in Europe. As you can see, those members have BSL-3 and BSL-4 facilities across Europe. ERINHA is focused on highly pathogenic agents and is actually designed to provide an access to researchers in order to conduct projects that requires a large range of capabilities that are not available in a single infrastructure or a single country. So we focus, as I said, on high consequence pathogen with a priority on the WHO R&D blueprint. And we do so by providing an access to the member facilities. How does that work? Well, we have two ways. The first is through consortia funded by either the European Union or the European Space Agency, for instance. It could be any kind of funding bodies. But the second one is to individual users or groups of researchers through two kinds of ways. The first is paid for services, and the other one is the EU-funded transnational access programs that Claire is now about to present you.
Thanks, Dylan. Hi, everyone. So yes, as Dylan mentioned, I'll be speaking about the ISIDORe project specifically, which ERINHA coordinates. Dylan did the excellent presentation on this last year, so I know many of you might already be familiar with it, but for those of you who weren't at the UK ICN meeting last year, I'll go through a bit of the background and the way that the ISIDORe project works. So ISIDORe is funded by the European Commission through the Horizon Europe program, and it came about through a recognition that there were some lessons learned from the COVID-19 pandemic and previous emergencies, and that in general, one of the major lessons learned was that there was insufficient preparedness and response systems due to insufficient investment and planning and research and development. Not at all the fault of the scientists, of course. This is definitely the fault of the funding bodies and the more public perception of the importance of this. So ISIDORe's idea is twofold. So it's around responding to the COVID-19 emergency and also preparing for future epidemic and pandemics which may arise. So what we do at ISIDORe, we are a consortium. It's based around 17 different research infrastructures, which I'll look into in a moment. And what we provide is free of charge access to laboratories and services for scientists for their to help in the research for emerging or re-emerging pathogens. So we provide this service to the scientific community. So these 17 research infrastructures here on the screen, ERINHA, which Dylan presented on, we're the coordinator of the project, and we bring together these other 16 different research infrastructures. Many of them will probably be recognizable to you. And what we do here is we cover the entire research and development pipeline. This covers from basic the structural biology and imaging all the way through to clinical trials, and does include also social science research. And it's about the tools that are essential to achieve excellence in research and innovation. So most importantly, of these 17 research infrastructures, so as Dylan explained, ERINHA is a research infrastructure. We bring together our members who are access providers or service providers. So then through the 17 research infrastructures, we bring together more than 150 different access providers who provide these services directly to scientists. So I know, for example, our hosts at University of Ljubljana are one of the access providers. And then through these 150 plus access providers, we provide access to over 300 different resources and services. So it's a really great resource for a range of different scientific services. How these services are organized into this catalogue of services we refer to, which is 9 different areas in general. There is a 10th one which isn't directly available to the scientists, and that's around the provision of the viruses and the reference materials. That goes, it's kind of a back channel service that goes directly to the access providers to provide the services. But as you see here, we have everything from analytical services, in vivo models, clinical samples, social sciences. So it's really that whole range. So how it works in general, I'm very bad at using pointers, so I'll just describe it from the top left. We're starting here. So the ISIDOre Strategic Advisory Board, this is made up of a number of different authorities in health and pandemic preparedness. So the ECDC, WHO, World Organization for Animal Health, CEPI, etc. They provide the advice which we use to design our different calls for proposals. So effectively, they look at what are the different priority pathogens that we should be focusing on, and then we design the call for proposals based on that advice. Moving down the left-hand side, the scientific community is then invited to apply for these calls for proposals. Once you apply, your application is reviewed independently of the project. And then once it's selected, you get linked up with one of these access provider partner facilities, and the service is provided free of charge. So just to be clear, ISIDOre is not a grant-making project. We do not provide funding directly to scientists, but we provide free of charge access to services, which can be quite expensive. So just a slightly clearer description of this process. There's a very short pre-application that you'll see on our website. We ask for a few basic details and about a 50-word description of your project. That just helps us to allocate you to one of these 17 different partners to see where your service would be required from. And then you'll be requested to complete a full application that's evaluated based on technical feasibility, so whether we actually have the capacity, as well as scientific excellence and ethics standards. And then that moves on to the access, which is either provided in person, so you may go to the laboratory. It may be remote, so the laboratory performs the service on your behalf and sends you results, or it could be virtually as well. It's important to note that you do remain the owner of the results of the service that is provided for your project. And we also provide the logistical and technical support, any training that may be required for this. So through to ISIDORE's activities so far. So we began in February 2022. Our first call for proposals was launched in May. So it was a very quick first few months to get this launched, and that was the SARS-CoV-2 call. So this was the emergency response call. That ran until February 2023. So we've now closed that call for proposals. We're focusing on preparedness, which I'll show you next. Once this project had already started, of course, then the Mpox crisis appeared, and so we very quickly moved together between May, launching the SARS-CoV-2 call in May. We then quickly pulled together an Mpox call, which was launched in July. That was a much shorter, more directed call. So these calls were both around controlling public health emergencies and really that response mechanism. But now in the later part of the project, we're more focused on the preparedness side. So the preparedness side, as I mentioned, with the Strategic Advisory Board, the 4 different general categories were decided, and we have a scientific strategy and a list of priority pathogens, which largely reflects, for example, the WHO priority pathogens into these 4 categories. So the risk group 4 pathogens, vector-borne pathogens, respiratory pathogens, and other pandemic-prone pathogens, sorry. So respiratory, which as this is a coronavirus-focused conference, may be more of interest. So that does include SARS-CoV-1, for example, MERS. It also, if you have a SARS-CoV-2 project that it's more pan-corona, so it's not just COVID-2 focus, then that would also come into the respiratory pathogens call. Other pandemic-prone pathogens is the disease X, the unknown, so looking at projects which would be effective for a range of unknown potential future pandemic-prone pathogens. So all of these calls are still open. There are some limitations, which I'll go into in a moment, and you can have a look at our website if you're interested in applying. And as I said, this is around increasing the capacity for the future. So in this kind of, are we in a peacetime yet? I don't know if we're just still coming out of the wartime, but the peacetime preparedness actions. So where we are to date, as I said, we're about halfway through the project. We've had, so I mentioned earlier, there's this small pre-application process, then a full application. So we've, up until this was the 1st of September, I believe, these statistics, we had 310 pre-applications, of which 144 people have submitted a full application. We have a fairly high success rate in that of those 144, 109 of them have been selected so far. And then in this pie graph here, you can see, because the SARS-CoV-2 was the first call, and the longest open and was specific to that one pathogen. We had 51 projects which are being supported by ERINHA for SARS-CoV-2, 14 for Mpox, and then you can see that the others there for the 4 different preparedness groups. But we have already committed 80% of the funding which is available. So this is where I mentioned that there are some limitations involved. We're trying to ensure that we have some balance across those, the different preparedness calls. We also know that some services, so your NHP in vivo studies, for example, could be a lot more expensive. We may already be reaching limits there, but some other projects are less expensive. There's also the time limit. So as a EU Horizon Europe project, we do have a strict time limit. So we need to make sure that these activities are completed within the timing. So that's just an idea of some of the limitations, but we would encourage you to reach out and we can discuss any requests. So we'll go on a little bit to some of our success stories so far, and I will just caveat this with a small apology that neither Dylan nor I are scientific experts ourselves. We come on the project management and communication side, so apologies in advance if we don't do the best job at explaining this. But we'll present a few different user cases that we've had so far. So this first one here was Dr. Markot at the Karolinska Institute in Sweden approached ERINHA and is looking at a study on the binding region of an anti-SARS-CoV-2 antibody, looking at the Omicron subvariants, the different subvariants. So you can see his question here is, what are the binding properties of the MAB19N01? So then he, through EU OpenScreen, which is one of our research infrastructures on high impact screening, was linked up with the IRB in Switzerland. And so this is really where this European increased capacity happens. This is all transnational access, meaning that it's a Swedish user who is being provided a service for their project by a Swiss institute. We are EU focused, but we have partners outside of the EU, across Europe and even outside Europe. So the IRB then performed the work on behalf of the Karolinska Institute. You can see here the 4 different steps of inhibition, epitope mapping, computational simulation, and then the kinetics parameters. And what was found is that the MAB19N01 does compete with the ACE2 binding to the RBD of the SARS-CoV-2 spike protein, that it neutralizes all the SARS-CoV-2 variants that were tested in the study, and that there's a nanomolar efficacy. So then after these services were provided back to the Karolinska Institute to help advance their project, they have now, if that's working, there's an article which has been published in iScience. So I would encourage you to go and have a look at that if you have, if you're interested in that particular study. And that's just an example of how, yes, the ISIDOre project through these services is helping to advance the study of particular users. I'll let Dylan present the next 2 user cases.
Thank you, Claire. So I'll try to do my best. So the next user case story we are presenting today is that of Angelina Svetlova, which is a PhD student in the Max Planck Institute. And her work is actually focusing on the long-term impact of SARS-CoV-2 on the cardiac function. And so via ISIDOre, she applied and she got an access to contrast imaging and was able to quantify the structural changes actually caused by the infection, including in ventricular wall thickness, lumen volume, and the state of the heart valves. Then she took a high-resolution computed tomography scans, and it took the analysis to a further level, giving her data to make a detailed 3D reconstruction, allowing them to zoom into precise areas of the pathology. This is some of her work, and as you can see, the image analysis is still ongoing and is should provide interesting results to give new insight into the effect of viral infection of the heart. So at the moment, as the imaging is still ongoing, there is no publication yet. But if you're interested into her project, I can redirect you to a larger, more detailed explanation on the website. And the last user case story I would like to present to you today is that of Dr. Dimikos, who is based in the European Biomedical Research Institute of Salerno in Italy. And his group specialized in the design of bioactive compounds. And he reached to ISIDOre in needs of specific structural biology to expertise and understand the mechanism of action of AT1001. So he received the free of charge access to X-ray crystallography pipeline and performed these studies to reveal how exactly this AT1001 binds to AMPRO, and informed then Dr. Dimiko as to how the drug design should be performed to refine generating improved second generation peptides. So as you can see here, it resulted in the characterization of compound with both promising actually anti-SARS-CoV-2 activity and very low cellular toxicity. That actually has been published, his work has been published in the European Journal of Medicinal Chemistry, and I encourage you to go through the research if you're interested into it. There will be all the details out there. And that concludes our presentation of the ISIDOre project and the user case stories. I hope it was clear enough, but again, if you have any question, we're here to help you. And also, if you would like to access the papers we showed, please feel free to contact us. Thank you very much.
Well, thanks very much, Dylan and Claire. Can I just ask, what's your kind of position if somebody would apply and you know, it's suggested they need more results. What's the policy on reapplication if they get more data?
So if they applied and received a service and then wanted to apply for a second service, is that your question?
No, I mean,
Sorry, or-
maybe the same service, but they go back in with more data.
So I mean, it's also not a just a one-off service provision. So once the user is matched up with one of these service providers, there's definitely a consultation that happens there as well to make sure that the user is getting what they need and that the service provider is fully understanding the project. So it can be an iterative process. In general, I will say that we would aim to make sure that this funding has the widest impact possible. So we would, in general, prioritise first-time applicants, but that doesn't mean that we wouldn't fund a second-time applicant. That would just be based on part of this evaluation and the technical feasibility that's done to ensure that we're finding that balance between breadth of service provision and also ensuring that we have some really useful results for the scientific community.
Okay, thanks very much. Any questions from the audience?
Hi,
Miles?
sorry. Thank you. Hi, thank you for your presentation. It's really interesting. I noticed that in the slide where you had the sort of different types of services that people could apply for, there's in the corner, you've got social sciences and epidemiology. So I'm curious about the demand for social scientific expertise and support, and if you had any examples or use cases of that. I'd be curious about the kinds of maybe social scientific expertise or support or questions that would be that ostensibly mostly, I would imagine most of your applicants are maybe from a more biomedical background that they would be having. Thank you.
Sure, thank you. And thank you for your presentation yesterday on the social sciences. It was very interesting. So the social sciences services are provided by SONA Global, which is a network coordinated by Institut Pasteur. For example, I mean, I don't have a specific user case for you, but some of the examples of services that have been requested is often around advice and assistance for example, designing of a fieldwork type survey or questionnaire, so helping with the methodology design there. They have a lot of partners so both service providers and users, often in global south countries. So there's been a lot of work, especially around some of the vector-borne diseases, and for example, looking at community responses. So, you know, there would be service provision around community responses to vaccine uptake that you are obviously working on. What I'd advise as well is go and have a look at the website, because once you can click on this and then you can go in and we have a more detailed list within that category of the different subcategories of services, and we can put you in touch with SONA Global contacts to look through what might be a suitable service.
So you just answered my question, and that is, for example, I was running a vaccine for Lassa fever, and I needed a partner to do Lassa fever neutralization assays. Is there a search mechanism, or you just click on one of the buttons you think is relevant and go from there?
So yes, so when you click on the button, so I'll do one that I know better. Once you click onto the in vivo models, for example, you'll then get the sub list, and you'll get the you know, whether you're looking at a mouse model or through to an NHP model. And so there's a lot more detail within this. So you can definitely look through that. We also do encourage you just to reach out via the contact email address if it's not particularly clear. We do have a form you can fill in if there's a service that's missing that you would like to see added to the project, or maybe it's just that we haven't quite clearly communicated it enough for you. So do encourage you to reach out if you also can't necessarily find the service that you're looking for, because I can't guarantee you that we have it, but we have a lot. So yes.
Right. Thank you very much, Dylan and Claire. Thank you.